Long-duration space missions have become an increasingly important issue as space agencies look to send humans to the Moon, Mars, and beyond. Engineering, physics, and mathematics are important in terms of transporting humans to another planet, but when it comes to keeping the passengers alive, biology becomes a priority. Bacteria and other microorganisms play crucial roles in how life exists on Earth because they are present in most biotic processes, including during human bodily functions. Therefore, to maintain human life in a spacecraft, it is necessary to consider the microbial organisms as well. The International Space Station may seem like an unfavorable environment for life, but looks can be deceiving. The ISS has been orbiting the Earth for nearly 20 years, far away from the reaches of Earth's biology. Yet, humans have had a consistent presence on the vehicle since its construction, contributing to the deposition and growth of bacteria on surfaces on the ISS, some invisible to the naked eye. Bacteria can come in all types. Some are beneficial for human health, some are virulent, and some can be relatively pleasant on Earth but become harmful when placed in a stress-inducing environment such as space. As Willie, Sandman, and Wood 2019 point out, factors such as nutrient starvation can lead to a radical expression of genes within a microorganism. Contamination in an enclosed space can become dangerous for the astronauts on board, and raises concerns for space missions requiring longer stays in such an environment. Balancing healthy levels of bacteria while combating dangerous microorganisms is crucial for the longevity of the ISS and future spacecraft. Certain bacteria respond to environmental stressors through various survival techniques, such as spore formation or halting growth, according to Willie, Sandman, and Wood 2019. These coping mechanisms enable organisms to persist through harsh environments while also making them resistant to traditional antimicrobial techniques, and potentially more virulent. In fact, some microorganisms capable of altering morphology or slowing their metabolism in the presence of a stressor become more harmful than their unaltered versions. In a study performed by Venkateswaran et al. In 2014, a vacuum bag of dust and lint was collected from surfaces of the ISS and returned to Earth-based labs for testing. Contained in the sample were various debris, including human hair, food particles, plastic, nail clippings, and skin fragments, according to Mora, Paris et al. In 2016. The biologicals were plated on various agar types and subjected to many tests to determine the identity of the microorganisms present in each sample. Among the microorganisms typically associated with humans were spore-forming bacteria and fungi. Staphylococcus was abundant in the sample, but since it is a common bacterium found on healthy human skin, it was not a large cause for concern. Members of the Bacillus species were discovered in the sample, but not grown successfully. Speculation surrounding the lack of growth exhibited by the spore-forming species suggests that the bacilli found in the ISS bacterial strains might be accustomed to different nutrients than common Earth-based bacilli, according to Venkateswaran et al. In 2014. Even though certain strains of Staphylococcus and Bacillus can be harmful to human health, the study decided that the fungi in the sample were of more concern than the microorganisms. Aspergillus and Penicillium were among the fungi identified, and can be considered opportunistic human pathogens. Aspergillus and Penicillium release conida and hyphal fragments into the air, putting astronauts at an increased risk for allergies, fungal infections, and cancer, as noted by Venkateswaran et al. 2014, Kohler, Casadevall, and Perfect 2015. It was also reported that 23 astronauts experienced viral infections during their stay on the ISS, as noted by Mehta et al. In 2017, proving that microorganisms are a cause for concern. Terms such as sanitization, sterilization, and disinfectant are commonly used when referring to antimicrobial efforts, and they apply to the level of achievement attained in the removal of microbial life. Sanitization involves simply cleaning a surface and does not adequately remove all vegetative organisms. Disinfection is an improvement from sanitization because it can kill microorganisms. However, it lacks the ability to destroy spores. Sterilization is the most effective and the most difficult way to control microorganisms because it can kill anything that is alive, including spores. This was noted by Voorhees et al. 2019 and Willie, Sandman, and Wood, 2019. There are a variety of techniques available to sterility, such as filtration, chemical agents, heat, and radiation. Each method has its own strengths, weaknesses, and effectiveness for removing microorganisms from the area of interest. Mechanically, the removal of bacteria can be achieved through filtration. Engineers have already implemented a filtration system in the ISS to remove harmful particulates from the air while recycling clean, breathable air back into the vehicle. However, microorganisms that did not pass through the filter can accumulate on the filter surface over time and continue to grow. This was noted by Venkateswaran et al. 2014, promoting a buildup of bacteria in a region inaccessible to the filter. As evidenced, filtration cannot be the only antimicrobial method employed by the ISS, and chemical agents must be utilized as well. Chemical agents are commonly used in antimicrobial efforts, but they too have their limitations. Isopropyl alcohol and benzalkonium chloride were used to clean the surfaces of Kibo, an isolated module of the ISS, but they were not effective in attaining complete sterility because the targeted bacteria became resistant to its antimicrobial properties over time, as noted by Ichijo et al. 2016. This is a real-life example of microorganisms becoming resistant to cleaning efforts through mutations, as noted by Mora et al. 2016, and it also illustrates how difficult it can be to combat microorganisms on the ISS. Physical agents such as heat and radiation are effective sterilization techniques. Heat can come in the form of dry heat, such as flame, or wet heat, such as steam. Exposing glass to open flame or leaving it in an oven overnight will sterilize it, but this can be time-consuming. Steam is more penetrative, which is why autoclaves are commonly used to sterilize laboratory equipment. Spores can be killed within minutes of exposure to steam in an autoclave. Heat is effective because microorganisms are incapable of regulating the temperature inside their body and become highly susceptible to changes in temperature, as noted by Willie, Sandman, and Wood 2019. However, it is not an effective sterilization method for all bacteria because some microorganisms are thermophilic, meaning they thrive in heat. Additionally, humans are highly susceptible to extreme temperatures, so any attempt made on the ISS to kill microorganisms would also put the astronauts at risk. Another physical agent to consider is radiation, which can come in the form of ultraviolet, known as UV, rays, or gamma rays. UV radiation consists of a long wavelength of electromagnetic radiation and is typically used in cabinets or fume hoods to achieve sterility because it damages DNA, as noted by Willie, Sandman, Wood in 2019. This method is sufficient for surfaces, but will not penetrate the membrane of a cell. Gamma rays, on the other hand, are less efficient time-wise, but can sterilize anything due to their short wavelength and high frequency. Ionizing radiation immediately kill the bacteria, or employ its tendency to cause mutations in microorganisms, leading to a slow death. Radiation is already a concern for astronauts on the ISS, due to the lack of protection the atmosphere provides from the sun. In the radiation research analyzed by Nelson in 2016, it is noted that the external surfaces of spaceships contain properties that shield the internal region of the vehicle from electromagnetic radiation outside. Therefore, both large organisms and microorganisms are safe from the harmful rays surrounding the ship, except for random galactic cosmic rays and solar particle events passing through, according to Nelson 2016. Effective sterilization of the ISS proves to be an ongoing challenge in terms of balancing the elimination of virulent bacteria with the retention of human life and associated microorganisms. But a greater understanding of the microbes on the ISS will aid in creating more viable cleaning options. It is inevitable that biological growth will occur in spaceships inhabited by humans for long periods of time, but regulation of bacteria can improve the quality of life for the astronauts on board. Future human exploration into space hinges on the success of maintaining a healthy living environment for astronauts, and an understanding of the microbial world is a step in the right direction.